See? Okay, so I think we can start this presentation, this Friday talk today. We are very glad to have here Rafael and Ramiro to explain about this project. It's a new European project. Colomban was supposed to also be presenting this project, but he had to leave yesterday, so he cannot attend. He's not here today. So during this week, perhaps you have seen it, there has been a lot of people from many different places. There was a meeting from the BIOcean5D. This is a very big project that started 1 year ago, or half a year ago, perhaps? 2 years ago, wow. So the project started 2 years ago, and I asked them, since they were here, that that would be a nice opportunity that they can present the project and perhaps a bit of the results. For that, we have Rafael Siano. We know each other for more than 20 years. We're talking today just now. He did the PhD in Naples and then the postdoc in Roscoff, and he has been participating in the exploration of biodiversity of protists, especially dinoflagellates, and now he's moving very much into paleo data from sediments to see how diversity has changed in the past times. And then Ramiro, we know him very well. He's a scientist here. He's a computational ecology and evolutionarist, which sounds very nice. And he also will present data from here, from samples in temporal surveys. So I think, thank you for being here, and the stage is yours.
Okay. I think, can you hear me? Yes, correctly. Thank you very much. Thank you very much, Ramon, for giving the possibility and the space for BIOcean to be presented here. As he said, I mean, we had a very great meeting today, during this week here. It was great. Thanks to Ramiro and to Caterina to organize that. So I'm in charge of presenting the project on behalf of the whole consortium and on behalf of Colomban de Vargas, as he said, that he has to leave. BIOcean5D is a European Marine Horizon Europe project that has started 2 years ago, and so it's about biodiversity in the ocean, as the name said. So the rationale between that is that, as you know, biodiversity in the ocean is threatened by human activities, by global variation and changes. So the idea is to measure these variations and to see how we can protect that. So the idea of BIOcean is to create and to have new data, integrated data and theories associated to economy and from the different kind of biological complexity from the cell level to the community level, and also integrated the human aspect into the assessment of these biodiversity changes and values. So that's why we integrate marine policymakers, but also the economical aspect and social aspect of preservation of this biodiversity. So it's we aggregate in this consortium different kind of people and expertises, from biologists to economists, from philosophers to economists, and social scientists and biologists. So more words to the preservation of our oceans and seas. The idea is to explore marine biodiversity over the 5 dimensions. That's why it's called BIOcean5D, the 3-dimensional space, the dimension of time, and the human dimension. That's the 5th dimension that we're given to the assessment of biodiversity. The principle is to get this data fair, to develop to explore biodiversity again, to produce new sequences and data, to get theories and new ideas about biodiversity changes, in an ecological and evolutionary dynamics and drivers manner, but also create, again, innovative and methods and protocols for economic and legal valuation of the value of the preservation of biodiversity in the oceans. So again, that's the idea, the space and times, the 4 dimensions, from organismal diversity, from viruses to mammals, from cell biology to community and ecosystem level, and integrating human aspects. Human aspects, so both, again, from the ecological, social, historical, and economical perspective. As you have also probably heard about, it's also linked to TREC. You have you had TREC here, so you've been shown how TREC was here. It's linked, not overlapping. BIOcean5D finance some part of TRECs and some actions. So the idea at TREC, you remember, is to sample from the inlands to the offshore waters to understand the connectivity across the biomes from the land to the oceans, from the sediments to the aerosols, this connectivity. And we also sample many different other ecosystems, holobionts, key species, like you can see here, for example, from benthic fauna to coral reefs, from deep oceans. We sample also in different ways. I mean, we collected different kind of data, not only genomics, but also non-DNA-based biodiversity assessment, microscopy, models, physicochemical parameters, and also, again, we have philosophers, economists, and social scientists that to evaluate the natural capital accounting of biodiversity preservation. It's a big project of including 31 partners. It started in it lasts 4 years. And it's a total fund of 17 million euros. And it integrates TREC in the consortium of TREC in this project. Here you have it's a European consortium, all the partners here. Of course, you are included. And that's the way we organize the work. It's quite complex. It's work package 1, which is really filling the gap into biodiversity changes, producing new data, and that's also where TREC has been implicated here and connected. Then we have a work package 2, where we analyze specific holobionts in the target ecosystems, so the holobionts of this ecosystem, and the way they changes. The link is the analysis of the microbiome of these holobionts in relation to changes. Work package 3 is to create a database uniformly structured across the project that is accessible to everybody, and it's also publishable. And then on the basis of this, so these two work packages create the data. The work package T assemble the data. Work package 4 gray is the theoretical parts where we develop new theories about biodiversity, and on the basis of this theory, we have some impacts analysis, both from the human perspective, so creating new bioindicators of marine health preservation, conservation, biodiversity, bioindicators inputs of pollutions, etc., or biodiversity change in relation to pollution, sorry. And then there is again this part where we evaluate the natural capital of the preservation of biodiversity, especially working, for example, plankton, how we can evaluate the interest of preserving plankton biodiversity from the economical perspective and social perspective. Here we have economists, social philosophers working on that. Then, of course, we have outreach engagement, a lot of outreach, coordination, and there is also another work package about ethics that we develop. Of course, it's a huge project. We are the leader of the project, for some scientific leader, is Colomban de Vargas in Roscoff, but the coordinator is Ensemble in Germany, and it's organized the work package, 7 work packages, I told, with probably you know some faces: Paola Bertucci, who is the responsible project coordinator of TREC, Fabien Lomar in France, Susanna Coelho in the Max Planck Institute, myself as work package leader of work package 2, Per Bork, Embol, Michael Retz in HZ, Tristan Cordier in Norway, Bruno Hernand in Ifremer, José Montoya in France, Ben Worzen in UK, Sacha Bourgeois in Nantes, Aranza Murillo is in Asti, Andrea Abreu here, and then it's from Embol from the outreach perspective. Of course, there are many people. I think we are more than 200 scientists implicated in the project, so, and many different postdocs and PhD students. So today, of course, we don't have the time to explain all the project, but we have decided to focus on 2 tasks of this project that enter in work package 1. One is the one that I lead about paleoecology, and the other one that Ramiro leads, who is the time series, TAX 1.2 and 1.3. So now we'll shift to the paleoecological part, the idea is to infer ecosystem evolution, biodiversity patterns from a paleoecological perspective over the Anthropocene periods. So it's also a big consortium, and the idea, the rationale behind is that biodiversity shift had been always studied using time series. These time series, however, are relatively short in Europe, but also all over the world, and the longest it's about 50 years old time. So it's not enough to depict biodiversity changes across a long time period, especially before human impacts. And so we lost information on past community, we lost information on baselines, assessment, biodiversity assessment before human contamination and pollutions. That's very important to analyze how nowadays how biodiversity has evolved in coastal ecosystems. And so we need long-term data sets, and of course that's why here come paleoecology. Paleoecology is the science of the past, the science that studies the ecosystem of the past. As you can understand, but this is in a broad sense. I mean, you have many different proxies and bioindicators of these changes in paleoecology. This is a nice review that shows you that, and so you can analyze this from different perspectives, using different proxies, biochemical proxies, biological proxies, chemical proxies, etc. And also important in paleoecology to study to which time scale you are referring to. So in paleoecology, many people working across at paleoceanographic level over millions of years or over thousands of years. So in my case, I decided to focus mostly on the Anthropocene period. So the last period when we have been called the era of humans. So when the human impact became important and relevant at the time scale of Earth history. That's the way it's been defined, which is also criticized. But anyway, this is the way we interpret nowadays the Anthropocene. So the idea is to collect sediment cores in marine coastal ecosystems, and I will be talking about two work I've done that led to inspired me to the work in BIOceans. So this probably you've been already know that I presented already this here when the story of the Bay of Brest. We collected sediment cores in the Bay of Brest here in the northwestern France in Brittany, different lengths. These cores had been analyzed by geologists, sedimentologists, chemical in order to validate before going to genetic analysis. Of course, they've been collected by with different vessels in coastal ecosystem, you consider very near shore. Then this course had been sliced centimeter by centimeters, and all precaution had been used to avoid contamination with modern DNA. And the idea that was, go directly to the results, probably you have already seen this kind of results. So what we demonstrated in this work that we collected the data over 1,400 years from the Middle Age to the modern time, and this is the variation of dinoflagellates across this time, analyzed, assessed by amplicon sequences on the V4 eDNA barcodes using ancient DNA. So as you can see here, what is immediately clear is that you've seen a change in community composition and dominance in this from the order Specialis in dinoflagellates towards the order Gonialacales. And this has been assessed statistically starting from the years 40s and 50s. Then in the 90s, we also observed statistically significant changes and variations at the genus level. I don't know if you can see here, but there is different color tones of red that had changed that show a different genera that became important towards the time. So we have already looked for the causes, potential causes of these changes, and we looked for contaminants, different kinds of inorganic and organic contaminants. These are families of, these ones, the first four are the families of inorganic contaminants, like, for example, mercury, nickel, etc. We found, for example, at 2 peaks of nickel and chromium, by the way, they're at the moment of the 2 World Wars, and especially the second one, the first one from the past, I mean the second one from the past, so the one from the Second World War, similar contamination that have been observed in 1945 at the Pearl Harbor base that had been bombed by the Japanese during the Second World War. And then what we observed after this contamination during the World War II period, this increase in contaminants of like mercury, and that's also this increase in contaminants of PCBs. Those contaminants are proxies of agricultural origins, pesticides even. So we associated this to the changes of landscape, and especially the agricultural political policy that have been done in Brittany, where I mean many eutrophication was arrived in the coastal ecosystem, and also so these changes in the cultivation in the region was also corroborated by data on palynology, on pollens, that shows these changes of the use of landscapes. So that's what we measure. So the accumulation between extreme events, which was the World War II, and the chronic chemical contamination that was to the intense agricultural activities of the region. So that's what we explained. Of course, we didn't explain the cause effects. That was just an association between the changes of biodiversity that we observed and these periods that corresponded to pollutants and human origin. So what was interesting also to show here that we didn't come back to the regional to the region of this community composition previous these contaminations. Even despite the fact that these contaminants, especially eutrophication, is now decreasing due to new policy. Politics in Brittany. So we didn't observe any resilience so far of these communities at the time scale that we observed, of course. So we evaluate this as an irreversible change that human activity had caused in marine coastal ecosystem, and especially in plankton, and so consider it to pre-industrial ecosystem status that was before, at least in Middle Age, before this contamination. So what was interesting here is to show that we have double cumulative effect of contamination within the Second World War activities and the eutrophication. So we were not able to disentangle the two, which was due to what? So we have decided to try to disentangle the effect of a contamination toward a change in community. We have decided to go to New Caledonia. New Caledonia is an overseas French territory that it's in the South Pacific, across in offshore Australia's waters, and it's a very important place for mining extracts of nickel. It's the 3rd country in the world for the exportation of nickel, for nickel mining. So it's a very important economy there, but it's the only economy that is there, more or less. It means that there is no very big cities, urbanization is not a real pollution, agricultural activities are not really well developed, but all the mining activity is really important. So here, the ecosystem, of course, is oligotrophic. So what we really wanted to observe is the effect of this contamination over the time in New Caledonia. That's what we did. We collected the core of more than 2 meters there. That's the way we proceeded. Some people that have worked with me here remember this story. So we sliced the cores every centimeters for hours. We collected different kind of proxies to evaluate changes. We have used specific precaution that's very important using ancient DNA. So for the moment we only assessed microkaryotic amplification. We use rarefaction of the data. That's very important when you compare ancient DNA data. We also focus on resting forming stages, use Pierre database. And so what we have found, first we were able to, in the basis of biochemical activity and analysis, aluminum and nickel here, we were able to separate the time in units, what we call time units, in 5 time units, and according to the different kind of inputs from the land over the coastal ecosystems. And so, as you can see here, these units are here represented on the rows, and this corresponding to different inputs of nickel from the land towards the coastal ecosystems. And we also were able to model the different kind of runoff of the sediments from the water catchments. So during the second period here, actually the inputs for nickel increased in the coastal ecosystem towards the volcano-sedimentary classical runoff that are present in the year. And this also, you can show that here there was an increase in the sedimentation rate here. So it means that in a very short time there was a lot of sedimentation. And compared to the other periods. So the landscape erosion changed the runoffs. Actually, they mined, they erode the mine, and without any control. I mean, these mining sediments arrived to the coastal ecosystems. And so we look for biodiversity changes, of course, in particular during the fourth to the second period. Here it's a very summarized way of looking at that. During the second period, this is period when the mining activity increased in time. There was this decrease in biodiversity that we estimated at 40%. So when the mining activity was very intense, I mean, there was a lot of erosion, a lot of sediment arriving. This impact caused 40% of biodiversity decrease in the systems. And especially, we have also observed changes in the composition, especially of chlorophyte and fungi and apicomplexa. That, I mean, chlorophyte and fungi increase, and apicomplexa, which are classically dominant in ancient ecosystems, are diminishing in time. Also, we observed what happened afterwards. During the first time period, that biodiversity increased again, but a big change occurred in the composition with fungi that became dominant in the community, yeah? Compared to the other periods. So we were not able to assign those fungi to a real origin, because the marker that we use is not adapted to assess biodiversity of fungi. It's 18S and not ITS. But we suspect, and now we are doing that, we suspected this fungi are from terrestrial origin. We will be assessing this more carefully with using another marker. So we also looked at the species dynamics, and we classified those species dynamics in 5 different categories according to models of species dynamics. And the different colors shows different dynamics. So species, for example, that diminished in time, species that increased in time, or species that had this concave dynamics over the time. So of course, and also species that arrived, some taxa of Catosaurus, of course, is, that we cannot define much better this kind of data because of the primer. Species dynamic changes as a consequence of mining activity, and we found some species. Actually, the species that we found are species that resist to this new activity. We didn't find a lot of new species arriving, but the species that are still here are most species that resist. So this one led me to think about the task 1.2 in BIOceans, which has been called the Paleocore project. The idea is to assess the impact of human histories over the last 200 years in Europe, looking at different kind of impacts from city harbors, agriculture, aquaculture, industry, and even the world. So what we have done is, in the frame of BIOcean5D, in a link to TREC, is that across the TREC track that you recognize probably, we collected the core, sediment cores of 1 meter, is in more or less 1 meter, in 15 sites. 15 sites that are here listed, and those sites correspond to different kind of impacts that we can define as harbor development, industry development, urbanization, aquaculture, etc. We, of course, sampled in Spain, but also in Bilbao, and for example, in Spain. So the question, the idea behind was how human activity in a broad sense has affected coastal biodiversity over the Anthropocene period. So in general, we want to try to disentangle the effect of a global environmental changes towards a local and specific pollution in biodiversity across the different study sites. If, for example, let's say, industrial development led to the evolution and emergence of specific communities in relation to another kind of, for example, of impacts. That's the same. Identify new specific assemblages and functional communities emerged after pollution. So if this pollution led to specific community emergent community, and describe the good environmental status before human pollution, like to use those data to assess baselines communities before the contaminations that are a reference point to evaluate other sites' evolutions. And also reconstruct the dynamics of some species across the time and latitude. Of course, also non-invasive species, species linked to human development and human activity development. The way we proceeded was this one, and I don't know if the video starts. No, but it doesn't matter. Okay, that's all. We collected cores actually even on small boats. Not here, it was a very big boat. With this small core, and that we collected more than 100 cores all the period, even by hand. And then we sliced the cores centimeters by centimeter for hours, 8-9 hours to collect all samples. And we collected at the end more than 10,000 samples overall across Europe. And we did it in a very interdisciplinary way, from meta-barcoding, meta-genomics, granulometry, dating, resurrection ecology, cysts and pollens, organic carbon and porosity, meiofauna, and contaminants. It's an interdisciplinary work. Of course, those cores will be analyzed by different kind of people and approaches, from geologists here that will reconstruct the sedimentology history of Europe, community shifts, both by meta-barcoding, meta-genomics in collaboration with Jopen in Poland, but also in collaboration with the University of Copenhagen in Denmark. Species dynamics and adaptation, both using resurrection ecology approach with Abo Academy in Finland, or with non-indigenous species dynamic with CNRS in France. And also contaminants analysis in France, colleagues in mine in Ifremer, both analyzing classical contaminants or emergent contaminant in the coastal ecosystem in relation, for example, to specific pollution that emerged during the 70s. That's where we collected the cores, the first results. So, I cannot give you much more results than those for the moment, but at least we know that the cores that we collected, these are the length of the cores, the radiography that we have produced so far of the cores, and these are the dating of the cores that have been sampled for the genetics. It is interesting to see, for example, in Barcelona, we did it back even more than the 19th century. We will be dating this for C14 to be sure of the dating of this core. So we have back to 4 cores that date back to the 18th century, 5 cores that date back to 19th century, and 6 cores that date back to the 20th century. But all cores covers the Anthropocene period, and all cores covers pollution that occurred in those sites. So I think we can really answer to the question that we had. We have decided to focus on specific topic. First, the impact of eutrophication, because the idea was to compare the same impact across Europe, to have an assessment of what this kind of impact have changed the biodiversity in Europe. So the nitrogen inputs, pesticide, etc. So looking at years 70s to the 90s. Then the impact of industrialization, in course that we covered the period of 19th century, industry development. But also we'll be looking at specific sites and specific events like the World War II, or for example in Naples, the explosion of Vesuvio, or the harbor constructions, the development of aquacultures, even across sites where this activity has been developed in time. So that's again the way the brown bars represent the Anthropocene period, and the colors in the questions. So we will compare the sediment samples of the different cores corresponding to the different questions. Where are now, for the moment, the extraction of DNA is being done by EUROPA. Joanna is here if you want to discuss with her. We will be focusing on eukaryotic primers and prokaryotic primers, also using the Fuhrman primers, which is one of the things we are trying to develop in BIOceans to standardize the use of those primers across different kind of approaches. And that we will be using that. So we are in the phase of analyzing those samples, half of those because we don't have enough money. And also we are starting the analysis of pollutants. So these are the real brand new results from 3 cores in Germany, in France, and in Bilbao. For example, in Bilbao is very nice because we can see that lead contamination decreased over time from the 1935. So we can really go through those changes in contamination and to look how biodiversity have changed across this time. Or we can study periods like here in France of contaminations that corresponding actually to the development of the town. And okay, we can see whether there is resilience of this ecosystem, because the Thau Lagoon is actually a study site in France for conservation ecology and restoration ecology. So we can see whether we can have resilience of the ecosystems. Or in Germany, we have specific contamination that were due to harbor development. There are peak of contamination that we also can go through that. Another part of the project is the outreach. So we are doing a lot of outreach, and we developed this toy, which we call the Paleo game. It's to teach kids and teenagers about the history of Europe using a core. And this, now it's developed. It can also be bought, and it will be used also for outreach activity throughout the project. Beyond all the analysis that we have been working together across the different collaborators. We are going more, from this project, I would say that there are branches that are developing about microplastics, specific contaminants, foraminiferous, lipidobiomarkers, and pollen. So we will, this is a project that generated other projects that is now ongoing. And we also analyzed what are the impact of this project in the future for the next time. Of course, this project completes the TREC exploration from the time's perspective, as you can understand. We created a consortium. So in Europe, people working on paleogenetics were very scattered, but now we created a consortium. We are now using the same methods, standardized methods, so we can compare our results in Europe. And we have more than 50 collaborators and 18 institutions that are working on this project. We are creating the first bank of past eDNA in Europe. So legacy samples that can be also used by other collaborators. So we would like to make it accessible to all of you that want to have access to the past history DNA. We wanted to study, the Anthropocene period, of course, in Europe, but this allows also collaborators like here, for example, to answer to local specific questions. There will be eDNA aliquots that will be delivered here and given here, and they will be free and open to everybody for use. And then again, there are new projects, more that we develop in the futures. Now we are more, I think 12 children that have been born from this project that are now developing in Europe. And then what I think it also is, this project allows to develop paleogenomics, which to me is the way to analyze the link between the land and the sea, We cannot disconnect the sea from the land, and the way, I mean, this is also written in many, it's the way to make this link is to study sediment cores and to try to analyze this, how coastal ecosystem has evolved from an holistic perspective, has evolved in relation to the human activity. And beyond everything, I want to say that it was a human experience. It was great to meet all of these people. There were people that were essential, that worked with me, that extraordinary people that work in the, I don't know if people are in the room, Mattia and Sandra, beyond Ramiro and Eva, that helped me a lot. That was really, really nice to work with them. So it's an incredible human experience that we are living. And we have excellent team, both in the Basque countries and in Catalan countries. And so, with this, I thank you very much. Again, it's a big consortium that they become from the BIOceans of 18 partners in 10 countries with more than 50 collaborators. And a special thank, of course, to Ephraim Amble and to TREC that funded and allowed this project. Thank you.
Thank you, Raffaele. Okay, let's see how much time I have left. No worries. I will try to. I will try. No. I will try to adapt. So yeah, I will talk a little bit on the work we are doing in time series. Of course, this work started before TREC. It's not. I mean, it falls within the umbrella of TREC, but started before. And it falls within this objective, which is comprehend whether marine microbiomes in comparable location display similar or different dynamics over long temporal scales. And this story started a few years ago, I think 2017, with Pierre Galland sitting and having a beer in the ISMI in Leipzig, asking, is these microbiomes that we are investigating in Spain, in Blanes, and in France, are the same thing or not? But just to highlight that, I mean, these time series have been running for much longer times, and it fits well within this project, let's say. So one thing that we've been wondering is how connected are these two sites, and this is, well, thanks to Sergio, who is sitting here. We use this app to try to see which currents might be connecting these two sites and, well, how this could affect the connectivity of microbes in these two locations. We know that these two places, Blanes and Sola, I mean, they are quite similar in terms of environmental variability, despite some changes in salinity that we can see here, for example. So in principle, these are comparable locations. The questions that I wanted to address today are these three. I mean, for these microbiomes, these two long time series, we wonder where they feature similar diversity patterns, if they have similar rhythmicity and synchrony in their microbiome, and where they harbor the same populations. So I will start with the first one, and I will try to be quick because, I mean, several of you know this already, and so I can fit in the time. So no worries. So we have, well, these two locations again, and then we selected 7 years of data from both of them. We were mostly focusing, in this case, in metagenomes. And basically, what we wanted to do is to say, well, let's compare these two locations, these two microbiomes at different levels of organization. So we look at them at the gene level, at the species or OTU or species level, and the functional levels as well. So the first thing that we saw, and here is data from Blanes and Sola, and this is time, and this is the, let's say, bright curves of difference at the whole metagenomic information level. We start to see something that we expected a lot, which is seasonality. I mean, there's a rhythm in the differentiation of the communities. Again, this is all metagenomic information when you take the bulk of information there. Same thing pop up when we look at taxonomy. In this particular case, is MTags, could be OTUs or species if you want to call them. And also when we look at genes in this case is open ring frames determined from the two locations. We see the same pattern and function. So we find a clear pattern of seasonality. Then if we do a clustering of these dynamics, we see some potentially seasonal clustering in both locations. I mean, we have samples from Blanes and Sola clustering together, which is something that we wonder where that could happen and was expected. So they tend to cluster together at different levels of organization. Again, we have all metagenomic information, taxonomy, open reading frames, and functions. We have samples here, for example, from winter and spring clustering together, for example, and then summer and autumn as well. This is a pattern that is popping up also in other data sets from metabarcoding, for example, and so on. So then we thought, well, I mean, let's compare the two locations at broad metabolic levels. This is Blanes, this is Sola, this is level 2 of the bright category in K. Well, this is just a technicality, let's say. So when we look at general, let's say, metabolisms, we don't see a difference, really. I mean, this is quite broad. This is what we expected. And we say, well, I mean, this is, even though there is seasonality and so on, I mean, these metabolic pathways behave in a quite stable manner. But then we start to dig in, and of course, when you dig in, you start to find things, well, usually. So we started to look at the coefficient of variations of some metabolism or pathways. And this is a different level of the category. And here we plot those categories which have the highest coefficient of variation. And we have things like flagellal assembly, bacterial motility, and so on. And we have the 2-time series here. And one interesting thing we find is that things that are quite variable in, for example, Blanes, they're also quite variable in Sola. So there seems to be a pattern going on. Then what we did is try to see the dynamics of those specific pathways. And here, I mean, the first 4 ones here are indicated here in this other plot. So you can see that there is a seasonal variability in these specific pathways. For example, bacterial motility, flagellal assembly, and 2-component systems, transcription factor, and so on. These are here the samples ordered according to time, and we have the 2-time series with the different colors. So there are functions, or broad functions, that show seasonal variation. We still try to find out what that means. I mean, we don't have an answer why bacterial motility has such a seasonality, but we're still investigating that. Then we a comparison between the 2-time series. Well, again, we have several samples from Sola, from the picoplankton, over 7 years. And we also compare against the global ocean to have a broad perspective. I mean, try to put into a broader view, I mean, the patterns we see in this 2-time series. So here it's just a simple classic accumulation curve. We have M tags, and we have Blanes in blue. Let's focus in Sola and Tara. We will ignore EMOs for now. We can talk later if you like. And we have number of samples here and the species accumulation in this particular case. So what we see is that Blanes and Sola we detect more diversity than when we compare with samples from Tara, which are comparable, let's say same size fractions and so on. Now, when we change to detected open ring frames, we have a change in the pattern. We see that we detect more genes than in the 2-time series. When we go to functions like keg and eggnog, well, curves accumulate in all cases. So I would say that the main differences are related to the number of genes predicted and number of taxa. And this could be related to, well, we have different theories. One could be the accessory genes and so on, but perhaps it could be that the variability over time, over the years, is higher than what we can detect in the open ocean. So now a few take-home messages of this part. So we see that the seasonality in Blanes and Sola, that there is a clear seasonality at different organization levels, OTU, gene function. We see comparable patterns of seasonality in both time series. We see limit functional turnover which was expected, except some specific functions. And we see also comparable richness patterns over 7 years in the 2-time series, and a higher taxonomic richness in Vargas and Sola than that observed in the global ocean, but lower richness of all, which is something that we are still interpreting. So now I'm going to switch to the second question. Do they display similar rhythmicity and synchrony? And this falls in the work of Sergio. So a lot of what I'm going to show here is done by Sergio for his first paper of a PhD. So we were wondering about how synchronized are these 2-time series and about the rhythmicity of the microbiome in the 2-time series. So this is a kind of conceptual framework. We can have taxa with high rhythmicity, with low rhythmicity. Here are 2 communities which are connected. These dynamics could be synchronized or not, and so on. So the first part, we are looking at, I will come back to this one, don't worry. We look at the rhythmicity and try to quantify this rhythmicity in the 2 locations. Well, I mean, I'm not going to go into details because there's no time, but basically we have the 2-time series here, and then we try to quantify how many of the OTUs in this particular case are rhythmic. And what we see is that about between 50 to 60% of the OTUs in the 2-time series show clear rhythmicity. And these were represented about between 68 and 77% of the total abundance. So we could say that there's quite a bit of rhythmicity at the OTU level in these 2 locations. Same figure, but now with open reading frames or genes. We see that between 40 to 54% of the genes were rhythmic in both locations, and that they represent, well, between 60 and 70% of the abundance. So the majority, let's say, or a good fraction of the genes show clear rhythmicity. And now we move to the functions. Again, I mean, don't worry too much about all these things, but the main message is that between 30 to 65% of the functions, depending on which database we use, were rhythmic. We have different database here and the amount of rhythmicity in the different functions. And this represented between 35 to 95% of the total abundance. So quite a bit of rhythmicity at different organization levels. Now we move to synchrony. And synchrony was interesting because, I mean, we wanted to see whether, I mean, the dynamics in these 2 locations are very similar or very different, and what that could mean in terms of ecology. So we were measuring synchrony, and basically this is the idea that 2 things change in a similar way over time. We try to quantify the amount of synchrony between well, different species and OTUs and genes and so on. So then again, I mean, I will explain this very quickly. I mean, what we see, I mean, here we are at the OTU level. Now we have the 2 locations, Vargas and Sola. And what we see is that in general, there's low rhythmicity at the OTU level. Here is the, let's say, the area of low rhythmicity. And basically, 66% of the OTUs had low rhythmicity, sorry, low synchrony. And that represents 61% of the abundance. When we switch to genes, in this particular case, same story, more or less, low synchrony at the gene level. So about 64% of the genes had low synchrony, and they represent about 46% of the abundance. And then again, functions, I mean, we have a similar story. Low synchrony at the metabolic function levels for the different database that we were using. So in general, it looks like there's low synchrony. So now I'm going to jump a little bit to something else. I'm going to do a stop here because I thought it was interesting to show this. But different to the previous patterns that I was showing, here we try to connect different dynamics with gene, let's say, patterns of redundancy, which means different taxa that can perform the same function, for example, and also patterns of dominance, taxa that are dominant and then influence the dynamics of, let's say, a specific function and so on. So in this particular case, what I'm showing is one specific gene. This is the nitrate reductase. And what we have here is that at the function level, we have something which is quite rhythmic, let's say. We have a high rhythmicity. But then there's a cocktail of taxa that contribute to that rhythmicity who are not rhythmic. This is the funny part. So there seems to be a pattern emerging at one organization level which is not based in something below. So this is quite interesting. The dynamics of this function is not reflected by that of its genes, let's say. And this is the, let's say, the conceptual explanation. We have a function at the higher organization level that cannot be explained by the dynamics of the things that are generating these dynamics. And this is the other scenario, which is, well, in this particular case, it's low functional redundancy. And remember, the redundancy here is, for this particular function, are different species that can perform that function, and it doesn't matter, in principle, that much who is doing that, but they are doing a particular function. In this other case, we move to low functional redundancy, so we have a specific taxa that are contributing to that function, and then the whole function is reflecting the dynamics of one specific taxa. So I just wanted to show this because I think it's a quite interesting result. I mean, there are more information I could show, but yeah, we don't have time. So just to close this part, I mean, we see that there's a broad range of rhythmic and arrhythmic functions, genes and OTUs, that display high, low, and negative synchrony in both microbiomes that we study. The low synchrony observing functions in and OTUs suggest a high idiosyncrasy in microbial dynamics in these two locations. And then, well, we see the functional redundancy and complementarity dynamics in lower organization levels might lead to rhythmicity at higher organization levels, which is quite interesting. And now I'm going to try to shrink in, I don't know, 5-6 minutes, the population part. So we also been investigating populations. When I say population, is let's say taxa that belong in principle, theoretically, to the same species. What is a species? I think we don't want to open that door here, but let's say that, let's have the species level in mind. So one interesting thing is that microbial populations are still a poorly understood dimension of diversity. I mean, we know quite little still about this, I mean, population of microbes. And understanding microbial populations could help us to better understand microbial adaptations and marine ecosystem function and so on. And this is just a figure we did as an example some time ago. This is, for example, one genome of one Flavobacteriales, one bacteria. And this is the distribution that we see when we look at the classic 16S, and that's good. But then if we open up what's inside this 16S, we start to see that we have some structure, and these are potential populations. We are not 100% sure if they are populations or not, but they could be populations, and some patterns that emerge. So we have two sub-questions, and this is work from Fran Latorre. The first is, do we find similar population variation in the two time series? And then we ask, how does the temporal population variation compare with that found in the global ocean? So we use an approach, I won't go into details, but basically we call it metagenome-based population genomics, which is extracting genomes from metagenomes and then using again the metagenomes to catch the variation at the nucleotide level, the single nucleotide variants. And then we have, well, a temporal data set, mostly is 15 years of metagenomes from Blanes, 7 years from Sola, and, well, from this year, 2008 to 2022, and we have a collection of 1,505 metagenomes, or MAGs, metagenome assembled genomes, from Blanes. And keep this in mind because these are the genomes we are investigating, we will investigate their variation, of course, in Blanes, in Sola, and in the global ocean. So for the global ocean, we selected some samples from Tara, from different areas, and from the same size fraction, in total 82 stations. And we jump directly to the results. We measure the FST. FST tells us the amount of population level differentiation for a given genome. So we have 1,505 genomes, so we pulled together all the FSTs that we extracted from these 1,505 genomes, and all those come from Blanes. Remember that. I mean, this is genomes from one location that we investigate in another location over time, and also in the global ocean. And what we see is that the overall FST, or percentage of population level diversity that we find in the two-time series over, I think, 15 and 7 years, is lower than in the global ocean. And this is kind of expected in a way, I would say. So then what we did is to try to categorize the amount of divergence. So this is the amount of divergence. Again, this is FST, and FST can go from low, moderate, high, and very high. And what we see is that, well, in the category moderate and low, falls most of the variation of the two-time series over 7 and 15 years, and a minor part of the variation in the global ocean for those specific genomes. Again, if you change the genomes, the story might change. But in this particular case, it's this for this set of 1,500 genomes. And, well, I mean, for the category high and very high variation, I mean, most of that variation is in the global ocean. Or in other words, let's say in the global ocean, we find several cases where the variation can be classified as high and very high, while this represents a smaller part of the two time series. Another thing that we did, and this is one particular example, is population differentiation over time. And this is interesting because it relates to the question of how much microbial populations are changing in contemporary timescales. We tend not to think too much on that, but we could think that perhaps in the last 50 years, and this is connected to Raffaele, the microbiome of the ocean could be changing or could display small changes. I mean, we don't expect perhaps complete new species popping up. That could happen. But in this case, we are thinking on population level variation. I mean, through changes in, let's say, dispersal or drift, or also evolutionary processes going on, I wouldn't say in the two-time series, but in the area. So in this particular case, what we have is the distance in days between samples. So we compare samples taken at different times, and we say how many days are in between, and the amount of divergence in terms of FST. And this is for one genome, and we have this done for several others. But this is an example of a typical pattern that we see. We see that, well, there is a kind of seasonality pattern, because we see that population differentiation is increasing and increasing over time. That is reflecting this seasonal pattern. But a more interesting thing that we are finding is that in some cases we have a trend in this amount of FST over time. So it seems that for some specific genomes, and this might not be the best example, but there are other examples with a much higher slope, we see that it seems that there is a clear signal of increasing FST over time for some genomes. What that increase means, well, we're still trying to determine what could be. It could be just population replacement, different in dispersal, or some evolutionary processes, including also drift processes. And this is, well, I'm arriving to the end. Sorry for the definition is not very good. But then we've been also clustering the FSTs of these amounts of differentiation between potential or between populations that are present in different samples. And what we have here is one example of one specific genome. And we've been clustering different samples where that genome is present. Well, we've been clustering with different methods. In this particular case, what we show is cluster by similarity, and then all the, let's say these are samples here, and those samples that are more similar in terms of the population that contains are clustered together. So what we see is that there are different clusters that pop up that could represent different populations. These are these colors. So these different populations seem to be, well, seasonal populations that we could perhaps relate some of these clusters to seasons. I mean, the seasons, we cluster those into big groups like are called autumn and summer and spring and winter. So they are mostly related to waters which are warmer or colder, two main stages in Blanes and Sola. So we also see that several potential populations cluster samples from the 2 time series. So it's not that we find 2 clusters linked to each time series. So the same population seems to be present in both locations, not always, but in many cases they cluster together. Here is Blanes, here is Sola in the 2 colors. As you can see, some samples are from Blanes and Sola cluster very near to each other. Well, we have other values which are related to, well, the intradiversity and the temperature. So, well, we are trying to link those potential populations to units that, well, could help us to understand why we have this in these different times of the year. So, well, the take-home messages that we have from this part is that do we find similar population variation in the 2 long time series? Well, we find similar patterns of genomic divergence in general, and we find shared populations between the 2 time series as well, which is, I think, quite interesting. And then regarding the question, how does the temporal population variation compare with that found in the global ocean? Well, we see that diversion at the temporal scale for the investigating mags, or genomes, seems to be lower than at the global ocean scale. And with that, well, I want to thank all the people involved, especially Fran, Sergio, and Pep and Ramon, who have been running the Blanes Time series for a long time, Lydia, Vanessa, and other people involved as well. Of course, Pierre Galland from Bagnols-sur-Mer, the funding, and also you for being here.
Thank you very much for these presentations and for these very nice studies of the temporal dimension in different categories, over sediments and on long-term seismics. So we have some time for questions. You are not the first, no.
Thank you. I was wondering, just because you mentioned these 5 dimensions and you focus very much on time series, but particularly with Ramiro's work, I was wondering, is there any study, because I think it's most on surface, right? Whatever there is something at deeper layers of connections, because also I was wondering up to which point some upwelling, downwelling phenomena could be related to changes on that.
So, yeah, the original idea with, and we tried within BIOcean5D was to sample cores from Blanes, but we hit a rock very quickly. So the core break. I mean, it would be super interesting to link. I mean, the dynamics in the, in the plankton over time with what we see at larger temporal scales. At the moment, it hasn't been, I mean, we are trying to do it, but we need to find a good location in order to do this linkage. We were talking about this morning, which could be a good time series that, that shows a big overlap.
Yeah, When we sediment course, we are faced to the sedimentation rates. So of course, the way, yeah, one layer could in some cases represent 10 years, in some other cases 1 year. So you have to find the time series in a place where the sedimentation rates allows to do that. So we are thinking of that in Europe.
And well, just another particular question. I was wondering, you mentioned what you have done in New Caledonia. Do you know if someone are working also with microbenthic communities? Because I suppose that mining has to be a lot of impact, not only on microbiome, but also in other organisms.
You mean in paleoecology, in paleogenetics?
No.
In
general, that someone could give some ideas how this mining has effect, because of the oligotrophic, you mentioned there is a lot of sediment, transport, extra, so on, and I'm supposing that is changing
of course we will have the DNA that will allow also to go through macro invertebrates, for example, etc. We also wanted to, it's a complicated story, but Ramon, we have to discuss about that here, whether from the parasites of the past we can reconstruct the host of the past. Something that we can also do. We will be working on meiofauna. It's not microfauna, but we have samples to go into meiofauna because it's enough material to get through the analysis, also by microscopy. And then, yeah, the DNA. Then we have colleagues in French Polynesia and in New Zealand. They are doing the same thing to try to link the South Pacific zone by paleogenetics as well.
Yeah. Okay, thank you.
Thanks for the talks. Every time I think about these paleo reconstructions and things like you guys do, I always wonder about of course, I mean, there's one core, there's several, 2-3 cores, and everything is like, well, and I measured these 3 variables, so I intend to explain the changes in the core by 3 variables, but I have 1, so there's statistically like a nightmare. So is there a way you can think of experiments in which you could actually test that whatever the bombs of the World War actually impacted and how? Or this is just to give an example of something that you were telling us. So is there a way that we could incorporate experiments into this type of research so that it could actually help you build cases that are statistically powerful?
Yes. What we were discussing yesterday at a nice tapas bar. First, for example, you can start from pristine environments, pollute the environment, and look if in the sediments you can see the evolution that you see in the past, for example. So that's a way. They did it in New Zealand, for example, and they showed that if you take a small amount, you eutrophied artificially the zone, the evolution follows the evolution that you saw in the past. So that's the first example. Another example would be, but it's not really an experimental part, it's more modeling, associated modeling to the paleogenetics. Imagine a zone that is connected by currents, and to see whether those currents, the connectivity of the ecosystems represents the information that we have in the past. Whether two ecosystems are connected, show the same things in order to have more science. But we are also thinking at this. Imagine that in paleogenomics is not a really old science, so it's really becoming more and more widespread, and the idea is to make the efforts together to do that. But we have some ideas, yes. If you are the ones,
I have a question about paleogenomics also. I think it's fantastic that you are able to identify the genetic signatures of different times in the course. That's fantastic, but how much is these genetic signatures is telling you about the community that was in the past?
That's I would like to discuss with you, whether it's, for example, one of the typical signals that we see in many courses is the presence of gregarines. In many courses we see that. And sometimes very few ASV, actually. We discussed with people doing studying Gregarians, yeah, and we were thinking about reconstructing, as I told you before, the community of the past using, the parasites that we are. If you, if we infer, let's say, that one single ISP is parasiting one single host, I mean, this is something, tricky, you know, if there is no, that's a way, and that's also, I mean, paleoecology is not perfect. We have to assume that the signals that we have in the past are not completely overlapping with what we see in the plankton. So there is something that degrades, that we can measure that anyway. But there is also, we mostly look at resting stages to be at least in a phase that. And then but what we think is that whether you see once we see differences in the sediment core, something should have happened in the ecosystem, even if it's not exactly the same that happened in the plankton, but something should have happened that caused potential change. So no matter the overlapping, we see changes,
Hi, thank you for both talks. They were great. So I have a question for Ramiro. Perhaps you mentioned it and I missed it, but what about the unique taxa? So taxa that are exclusively found in Sola or in Blanes, and they are present and recurrent, because they could mean...
There are some.
There are some?
They are only found in one place, and this could be related to some of the idiosyncrasy
of the local...
I mean, these locations, the whole thing started to, with the question how similar they are, how much information or species, genes, function do they share. So as many answers in ecology is a little bit of everything. So this is the...
So the proportion of shared, for example, is it very
The proportion of shared,
high?
well, depending on which level you're looking at. It could be, OTUs, genes, or open wheel frames. So if you, for example, if you look at the functions, they share many functions.
A lot,
But if you look at the OTUs, I don't have in mind the exact number, maybe Sergio remembers, but it was relatively low percentage, not very low, perhaps 30% or something like that. I don't have the number in mind, but we have it.
Because number. Those also, I think they can provide some information regarding the specific conditions.
So the two locations are, in general, we say, 150 kilometers apart. They share a similar environmental variability, but then they have differences. They have differences in, for example, there are storms happening in Banyuls which are not as big, probably, with the ones that occur in Blanes in terms of the, for example, water runoff and things like that. So there's a decrease. They influence the salinity of the location. But then again, where are the limits of the two communities? So that's something we wanted to explore. Are they part of a big patch that we could consider one thing?
Or perhaps the abundant can be dispersed and the rare ones cannot, or something like that.
And then there's another thing, currents, which we are trying to find out, how much they affect. Now, we've been looking at the northern current, which is what the plot that comes from simulation done here from the group of... it's a Cosmo application. Yes, Emily Garcia Landa. So we've been looking at that and trying to calculate, how much connectivity is in the two locations, because the same mass of water can be, first in Sola and then a few days later can be here. But it looks like there are also local effects in the two places. So the question is how much influence we have from those currents. This is still open. We need to ask the people, maybe someone in the room that can help us.
Thank you.
Yes, following a little bit on that conversation, when you show us about the two possibilities, the functional redundancy and the dominance of taxa, so I didn't get what is the first results. It seems that a high functional redundancy, right? And this is probably what it would be probably good to get like a more resilient community. So in case there's some taxa decay that is not a really, really dominant taxa, it will be replacement for other taxa that have doing these similar functions.
So there are different examples. So for example, the ones that I show, they are, two examples, contrasting examples. One, that there was low functional redundancy. So for example, if you look at the general function, it's dominated by one taxa or a few that contribute the most of the information to that function, let's say, or are responsible for the most part of that function. But the very interesting example is when you have, let's say, a more chaotic dynamics or regular dynamics, then when you put all those dynamics together, they lead to something regular. And yeah, in these particular cases, I mean, it's usually I try to think in a football team that it doesn't matter the players, you still have a football team. I mean, it's not that true, but, but then again, I mean, you know, different taxa can contribute and then the function can go on because at the overall ecosystem level, you need that function to be performed. So it doesn't matter too much who are doing that. But yeah, it can be quite important in terms of ecosystem resilience, I would say.
So we have time for a last question before we close, and winner is.
Thank you again for the talks. I have a question for Raffaele, and it's about...
It's a revenge.
It's a revenge.
I'm picking between my two chiefs. Yeah. No, I had a question more about the connectivity between the past communities that you sample in the sediment core with the current ones that we observe in the sea. So obviously there's a filter which is dormancy, but maybe you could also measure sort of the decay between the sea and what is stored in the sediment core and in the past with phylogenetic relatedness, and maybe this would display quite a huge...
You mean to look at taxa that phylogenetic associated that really we found, yeah, it's a good, I understand what you mean. Taxa that are phylogenetic associated that you found in the sediments like a trait that you can, yeah, well, that's what you mean? Is there a group, specific group that are associated phylogenetically that you can see in the sediments? That's what you mean?
Yeah, first quantify the extent of what is in the sea that is present, or at least was present as a phylogenetic signal in the sediments, and then see if there's a decay of this phylogenetic similarity a long time.
The major issue still in the sediments is the degradation of DNA. Yeah, still, whatever you do, it's the only things. And we have done anyway a lot of trying to compromise for example, you also in the sediment there's a mix. That's also answer to your question from the plankton community and the planktonic community. So it's a mix of the two. So it's very complicated to overlap the two. There are some signals, for example, gregarines are not so abundant in the plankton, but there are 80% of the protist communities in the sediments. So they are not really overlapping. But we can try to do that. You want to try?
Maybe.
Okay, so I think we close it here. Thank you very much for the talks.
Thank you for giving us the possibility. Thank you.